Okay, so welcome to the last talk of this series of Corpus Curiosum. You know already, many of you, because I see many familiar faces, but for those who don't know, we are Corpus Curiosum, as we just were discussing. This project was born in 2020. It is called Corpus Curiosum because, as the same way as the corpus callosum does in the brain, we are aimed at connecting early career researchers in neuroscience all around the world with the main goal of discussing critical topics and stimulating critical thinking in neuroscience. So about this session, it's being recorded, as you just got the notification. No worries, because the final Q&A, so at the end we will have a discussion. It's not going to be recorded. So the way how it works is that now during the talk, you can write as many questions as you want in the chat. Actually, we really encourage you guys to write questions. And then at the end, we will have a discussion with the speaker. So Maria will moderate this discussion, so we will read the questions. It's totally a safe space. There are no stupid questions. They are not going to be recorded, so please feel free to interact with us. And then about the length, whatever is the time in your place, we're gonna close this session in approx 1 hour and a half. Yes, so we said that already. Our main goal is to stimulate critical thinking in neuroscience through engaging early career researchers and fostering these nice discussions. Our values, our core values are, on the first place, we really appreciate the support to early career researchers. We embrace diversity, so we try to reach as many different places as possible, and includes people with different backgrounds, different locations, and so on. And we will always be accessible. We are accessible now. We will always be accessible. And that means that all our talks are available in YouTube for free. And all our activities, the talks, etc., they are always free as well. So this is us. We are Ines, Alba, Marie, and Marta. We are placed in different cities, different countries around the world. We are also in different stages of our career, and we created this project for you. We have been supported along the way by different organizations, for example, the FENS and the IBRO, so we are very, very grateful for that. We also received the IBRO Diversity Grant twice already, so we are also very grateful for that, because without them, this wouldn't be possible. And one or another initiative that we have is the Corpus Curious Mind School, this Curious Mind School. Yes, we created for a selected group of people. So instead of having a lot of people in this discussion, we selected some students, and then along different sessions, we discussed different topics, and then we could create also a nice community, small community with all of us. We are going to maybe do another of these Curious Mind School. So if this is ringing a bell, is sounding interesting for you, stay tuned, because maybe we have news about it. And now I'm going to introduce the next speaker, our speaker of today. He is Dr. Adhil Bhagwandin. He's a neuroscientist currently working in the University of Cape Town, and he's specialized in comparative neuroanatomy, particularly in the sleep-wake system. He earned his PhD also in South Africa. He completed after that a postdoctoral fellowship in the US and also in the Moscow State University. In 2014, he received a very important award from South Africa. And then his research is very diverse. He uses different techniques to study sleep across vertebrates, including very different species. And then he aims right now to investigate further the glymphatic system and its connections to sleep. So about today's talk, this is the talk. This is the title. So it is called Chasing Rainbows, Neuroscience Lessons in Africa. So Adil, thank you very much for joining us. And I will stop sharing and you can take the stage.
Thank you very much, Alba, for that wonderful introduction. I'm very glad to be here. Let me just share my screen real quickly so we can get started with things. Can you see my screen clearly?
Yes.
Okay.
Yeah.
So let me just put this on. Okay, so good afternoon, everybody who's sitting in the audience, and thank you very much for making the time to be here. I'm very grateful that to be invited to give this talk, and also to have you here listening to me this afternoon. I was in two minds about what to do for this talk. I thought instead of giving you a very lengthy, data-driven kind of talk, I would pick on something that's and try and talk about something that I is very close to me in terms of my own work, and to share my thoughts about it and parts of my career so far, and how I feel neuroscience is going, particularly in the continent that I live in, which is Africa. So I've titled this talk "Chasing Rainbows." For a couple of reasons. I'm using rainbows as a metaphor here, but um, there are plenty, and the image at the bottom just really does tries to describe or emulate what the diversity on the African continent actually is in terms of the number of faunal diversity that we have in this continent. And so, like Alba had just said, my career has been devoted so far to understanding different animals, and I found a lot of joy in that. I really enjoy animals, and I think that they have a lot to teach us in terms of how we understand the brain, and certainly the work that I've been fortunate to do in my career so far has allowed me to and made me more excited and more enthusiastic about wanting to understand brains of different animals in the ultimate goal of understanding how the human brain developed, and by learning from these other species, what can we learn about our own brains. That's the motto that I have. That's the goal of a lot of the work that we do. So for today, I wanted to share with you some of that. So I hope you will enjoy, and we'll have lots of questions at the end. So let me get started by two of my favorite quotes that I as I read different things. The first of which was Pliny the Elder, who said, "Out of Africa, always something new." And I found a lot of motivation, inspiration in this quote because from a lot of the work we've done, we've always found novel things in brains from African animals. And a lot of it can be related to how they survive in an environment, but also it asks an important question of how can this be related to humans, which is a somewhat of a translational aspect to it. And the other one that I did was from an author called A. Milner. Some of you might be familiar with it. He wrote the book Winnie the Pooh, and his quote goes, "Some people talk to animals, not many listen, though. That is the problem." And again, I find this to be very apt for this particular talk, because I feel sometimes that we neglect the value of different animals, and for reasons I will explain to you in a little bit. So let me start with a little bit of introduction. I put in this on here to show you the extent of which vertebrate brains vary. And here you see many different species, or at least examples from many different species, from the earliest recorded brains, and that being the lamprey. Let me just put my pointer on. Okay, so here you see an example of a lamprey, which is a really, really very old animal, and somewhat hundreds of millions of years old. And you see it moving closer across to various different vertebrate classes. So here we have frog brains, and here we have shark brains, and some reptile brains, the bird brain, fish brains, and then mammal brains. And what might start to emerge from you as you look at this is that there are certainly a lot of similarities across vertebrate brains that we've seen so far. And the colors in this actually do show that. Yellow, for example, is cortical representations across all different vertebrate classes. The blue is the brainstem. The sort of reddish, pinkish color up here is the cerebellum. And the point being is that there seems to be all these major regions of the brain seem to be apparent in all different vertebrate classes, which in the bigger scheme of things means something quite important, that these are very critical structures that have been conserved over millions of years, hundreds of millions of years, that is, to be able to allow an animal to function effectively in its and not just animals but humans too, but any creature to thrive and survive in its environment. So indeed, brains are very, very complex. We all know this, and this is why we're all very excited and trying to understand more and more about brains. We are starting to understand the complexities of the human brain more and more every day as technology evolves and gets so much better. We can pry different aspects of it to learn much more about just how complex it is. Many of you already know that the human brain consists of roughly 100 billion neurons, which in itself, connected to all of the other ones, creates about trillions of connections in the brain. And it is as advanced as technology is today, no supercomputer or no advanced computer can match what the brain actually does and the human brain actually does, which is quite a remarkable thing. We often take well, I won't say we as neuroscientists kind of appreciate it a little bit more, but the general public may not get that opportunity and that insight into understanding and seeing just how complex our brains actually are. But how does other brains compare to human brains? Well, they're just as complex in various different ways, whilst having similar areas that are common across all mammals and all vertebrates. They have specialities that allow them to thrive in certain environments. And I think that is the one thing that has always drawn me to comparative neuroscience, in that there's so much more to learn. There's always something different. There's always a different angle to look at, and that has inspired me for many a different year to continue the work that we've started, and continue the work that we are doing, in terms of understanding the structural integrity, the neuroanatomy of various different vertebrate brains. My next point is equally important. Neuroscience, as many of you know, is a vast, vast, vast field. It incorporates many different subtypes, disciplines. If you look at molecular, if you look at genetics, if you look at histological, there are countless different ways in which you can understand the brain. And again, that speaks to its complexity at many different levels. But the important thing is that a lot of people are trying to understand how pathologies in the brain operate, how they work, what is the mechanism of their function, things like Parkinson's or Alzheimer's, for example, which are very common neurodegenerative diseases that have huge impacts on how people live their lives. And a lot of these are starting to happen a lot earlier and without much explanation. So people have invested lots of time, lots of energy, lots of resources, trying to understand these various different pathologies and neurodegenerative diseases. And the work that they've done to this point has been pretty remarkable. But one thing that's been common, and one thing that I want to try and address with you as we move through this talk is, how does a young researcher or an emerging researcher try to understand the complexity of brain. How should one go about it? What steps should one take? What things should one look at? And should we follow what's been done before in the past? Should we not? Should we think about a different angle? That's kind of where I'm going with this talk. And the other question that I want you to think about as we are talking about, and I continue with the different slides, is how should a researcher or an emerging researcher attempt to model neurological pathologies? What should we look at? What angle should you take? How do you begin to address that problem? How do you begin to, maybe not even to find a cure, but to understand the mechanism better, understand the mechanism of the disease, understand how the interactions between whatever is in that that's creating that problem for that pathology in the brain to rectify that situation. So I really want you to think about these questions as I'm talking, and hopefully we can have a discussion about it a little bit. I have some of my ideas which I'm going to impart on you through this talk, but it's not to say that my ideas are correct. It's just how I'm viewing it at this moment in time. So I wanted to start off with something that's might seem a little bit controversial, but it's not really, and it's not an attack on anything. But what I wanted to ask is, or question rather, is a rat and a mouse model the best model to study the human brain? And I've put that there for now, just so you can digest a little bit. But let's go into a little bit of history about it, because we know for the history of neuroscience, at least in the 20th and 21st century, the rat and mouse model has been a gold standard for understanding brains and understanding different aspects of it, cognitive function versus physiological function versus, all these different things, molecular function, so on, so on, so. And typically what people do is they would do intervention studies and compare them to control, which doesn't have that, and then they have the transferring lines that are missing something or not, and many of you are pretty familiar with that already, so I won't go on for too long. But this has been the gold standard of neuroscience for many years, and this paper of 2008 by Manga and colleagues starts to show the kind of influence or the use of various different animals in journals from 1975 to 2004. So the colors here are represented in different shades of gray, from light to dark, over the different years. And you can see at the bottom the different vertebrate species that have been investigated. And we see a huge tower with the rat and mouse species. Okay, so I wanted to just highlight that to show you that it has been a quite favored species to understand how the brain works, and what people typically do then is translate those results into humans. So humans have been used, but for various different reasons, it's quite difficult to use that tissue or it's quite difficult to preserve that tissue if you're fortunate enough to have that. But it's not always the case. Over the years, macaques and other primates, which are very close relatives to humans, have also been used to understand the brain, understand pathologies, but many places have put restrictions on how and where, and actually stopped using primates as due to ethical reasons. So researchers don't typically have access to these animals. If you look at the others, you can see they're not used as often. Another species in comparison that has been used more often than others, other than the rat and mouse, which has been the cat. And the cat was very popular species in the sort of 1970s, and over time its usage has decreased. Now, I mean by time in the sense that how many pages of a journal are devoted to studies that revolve around rat and mouse tissue and brains, and this is what's represented on the left-hand side of this figure. So the situation is quite clear. To sum up all that I've mentioned is the rat and mouse brain has been favored for neuroscience, at least for the last 40 to 50 years. I want to, before go any further, just say that there's absolutely nothing wrong with that. But as all good scientists need to do is we need to sometimes question our own techniques, not take things for granted. And one of the questions then is, how sure are we that that the rat and mouse model is appropriate to study the human brain? And I found a paper by Perrin in 2014 that showed and described that 80% of rodent studies fail to translate well to humans, which is a bit of a problem. Again, I'm not saying that they're not useful species, but when you look at the data and you look at the amount of time, energy, and resources that are devoted to the rat and mouse model, then and if it's not producing the kind of results that, or the cures or the treatments, or that one is typically expects with that devotion of or that investment, we have to question whether is it an appropriate model? Is it something that we should be using for moving forward? And again, it's not that I'm taking away from the value of things that have been used and been proven by these models. And it's an open-ended question to maybe refine, find a different direction, contribute more to us understanding about translational work and how do we understand the human brain in a lot more detail, seeing as though we do not have good access to studying human brains and using human tissue as an example. So, which is where some of the work that we've done comes into place. And so you might ask yourself, like, okay, so it's easy to do rat and mouse studies because they're convenient animals to use. They easily you can get transgenic lines. You can get all these different things. You can keep them in animal housing units, and you can do a lot of good things with them because they reproduce pretty quickly. They have very short times in their gestation periods. So there's a lot of advantages, and I could totally understand why people would want to and do that. Again, I'm not criticizing them, but when I ask the question of why should we study other brains, like, well, what would be the value of studying other brains? Why should anybody consider it? I want to draw your attention to a couple of very fundamental studies in neuroscience that have come from the use of comparative or using species other than a rat and mouse. And the first of which, okay, well, I'll get that into that in a second, but let me just reiterate my point of potentially why rat and mouse might be not the best to study a human brain, because there's an 80 million year gap, evolutionary gap between the common ancestor of the rodents and the common ancestor of humans. That's an enormous gap. And many things can. So here we see a nice little phylogenetic tree that shows the separation between the mouse and the human, and they split roughly 80 million years ago. Okay, so when we're thinking about using rat and mouse models, we have to ask ourselves the question of, there's an 80 million year gap, and when we extrapolate information, are we extrapolating them in a pragmatic way, or in a way that reinforces leaps of faith? Okay, and I'm not again criticizing anybody who does this. I'm just pointing out the facts that do exist with this, and so in all of this, I wanted to really just uh, give you some examples of some critical studies that have been done over time that show that have been benefit from a comparative. So the first of which was understanding the reflex frog and or reflex arc in a frog. I'm not sure how many of you do electrophysiological studies, but this is one of the cornerstones of understanding how reflex arc works. And it came from an amphibian in 1833. Again, understanding dendrites and dendritic spines came from all of you, I'm pretty sure, know the famous Kayal, and his work in chicken allowed us to understand the distribution and the sort of anatomy, neuroanatomy, or neuroanatomical distribution of dendritic spines, which is fundamental to how we understand how neurons communicate with one another, the synapses in between, and many other physiological functions associated with that have been built upon that. And lastly, one of the most fundamental experiments in neuroscience was the action potential propagation work that was done using the giant or the squid giant axon. Here you see a couple of images of it and some of the results when you stimulated the axon of well, you see the axon here. It's quite large compared to what you would expect and to see in a human brain, for example. So my point being is that there are a number of instances. These are just three that I talked about from historical context that show us some value of how comparative and a comparative approach can benefit neuroscience overall. But I wanted to talk about a few more recent examples. And again, some of which you might be familiar with, others you may not. But I chose these three because I find them pretty fascinating. And the first of which is navigation in birds. How do birds that migrate navigate their way through their environments? How do they know where they're going? And were done in two different species. One was a songbird and the other one was pigeons. And they both showed that the bird brain can detect magnetic cues or the Earth's magnetic coil. And so in that way, birds that migrate are able to move from one direction to the other based on the magnetic or the magnetic nature of or the flow of on Earth itself. And that is I think that is just incredible how they are able to do this. So another good example and something I happen to have worked on a few years back. I'm not sure if many of you are familiar with the tapir. This is what a tapir looks like. It's a funny looking animal, but has a bit of a longish nose. It's not very tall. It forms part of the family called Perissodactyla, and Perissodactyla include things like the zebras, the rhinos. They're often referred to as the odd-toed ungulates. In comparison to the even-toed ungulates, where you think of cows and giraffes and the deer that you see quite often in Europe, Northern Europe, even in the savannah in Africa, the springboks and the kudus and all these sort of very famous. Those are all examples of even-toed ungulates. This is a little bit different, and these are odd-toed ungulates. They often only have 3 toes on each foot. And what's interesting about the tapir, though, is that tapirs are typically found in a rainforest type environment, which is very dense. You can see part of it here in the background. Sorry, it was the best image I could get off it. But the point that I wanted to make is that when you look at the brain of a tapir, what you notice immediately when you take the brain out is very large mammillary bodies. And you would ask yourself automatically the question, why? Why would an animal like this need to have such large mammillary bodies? And when you think about the environment that they live in, living in a rainforest and very dense environment requires you to have the ability to navigate with a lot of accuracy, so that you don't lose yourself in the forest, right? Finding food or getting back to your home, or indeed trying to evade yourself from predators. And the mammillary bodies, we know, are heavily linked in terms of its communication with the cortex, and the place cells in the cortex, and they function solely around trying to provide a sense of GPS, basically, right? Position in time, and it's all about navigation for the animals. Right. So that's something that's very interesting, because the next question from that could be, well, if the mammillary bodies are large, what does the hippocampus look like? Is there a correlation between the hippocampus and the mammillary bodies, for example, in an animal that lives in a very dense environment like this? So we haven't answered those questions just yet, but I've given you an example of how to extend these kind of things further. And those of you who do physiology again would perhaps know about Candel's famous experiments and to learning and memory, in what is commonly referred to a sea slug. Who would have thought? It's an invertebrate, right? And Candel had done a whole series of experiments in trying to understand the concepts of learning and memory. How do we go about, how do you show that biologically? How does one understand it from a neuroscience perspective? And his work in the sea slugs were fundamental in trying to understand this. I'm sure many of you know his textbook, which is somewhat of a stalwart and an absolutely fantastic book if you read it. But it's mostly part. I mean, I know I teach it when I teach my medical students about the neuroscience, and so Candel is quite a good reference for me. But anyway, learning and memory really gained a foothold through the use of sea slugs. And here's an example of what those sea slugs look like. Okay, so my point so far is that if we start to look in a comparative sense, it's not just only seeing differences in different animals, but rather often they can be translatable differences, or translatable features that we previously may have overlooked, should we only focus on one or two different species as the models for understanding neuroscience. But let me try and convince you a little bit more, and again, this is speaking very much to my own experiences on the African continent, and we know that Africa is a varied continent. It has many different types of environments, ranging from deserts to rainforests to savannas to it's basically surrounded by water. So the variety, the biodiversity in the African continent is massive. To give you some examples of this, on the African continent, for example, there are 1,100 mammal species, 216 of which are just primates. There are over 2,500 species of birds and countless number of different amphibians, I mean frogs and the likes, fish, reptiles, the most famous of which you probably know is the Nile crocodile, one of the reptiles, but there are a whole host of others, chameleons and the likes. So my point being is that the African continent is incredibly biodiverse. Yet we know an absolute, I mean, a very small proportion. I mean, understand a very small proportion of all of these brains. Here's a really good one. Sorry, I'm just backtracking a little bit. I actually forgot about this because I find this animal very interesting. It's the coelacanth. And the coelacanth is one of the earliest vertebrates. It's been around for hundreds of millions of years. And you often find them in the very, very deep waters around the coast of southern Africa. So this particular specimen was found around Cape Town, actually. And it's in a museum now, but it had washed up ashore and in a almost perfect condition. But my point again is that there is very little that is understood about many different animals and many different species of, and that makes me ask the question: if we don't understand a lot of these different brains, what secrets are they hiding that could possibly help us understand one another? Okay, but not one another, but the brains of humans or the pathology of human brains. And that's really what's driven me for many years, is trying to understand this neurodiversity on the African continent, and the differences in the neuroanatomy, the structural composition of the different brains, working out numbers, trying to calculate how many neurons are in this area versus that area, and comparing them to whatever other relatable studies that have been done about a similar thing. So again, I feel like I'm repeating myself, but I wanted to just really reinforce the point that there is a huge diversity in Africa, and very little work has been done to look at the variation and the brains in different African species across different African vertebrates. So you might ask yourself then, what does that neuroscience in Africa look like? Given that I shared with you that not a lot has been done in the comparative. And so what we do find is that there are pockets across the continent that focus on neuroscience, largely in the north of the country and in the south. South Africa has a few different universities and a few different research labs that are focused on neuroscience itself, and I'll get into some other details in a second. Egypt is another very big country, Tunisia, Morocco, and then West Africa, particularly Nigeria, is also becoming quite a center for neuroscience. But when we look at the types of neuroscience that's done in those, it's largely things that revolve around the medical facility or faculties, understanding things like diseases, neurodegeneration, injury. If we look here, structural variations, cognition and motivation, emotions, and the likes. So again, sensory and motor systems, something also focused on. But the point is, a lot of this has been done using, with the exception of parts of Morocco and South Africa, a lot of the work so far has been done in rat and mouse species. Okay, again, I'm not criticizing, just pointing out what it is. So what are the challenges then of actually focusing, working in neuroscience in Africa? And when you work as part of your career here, you start to notice some common things that make it quite difficult. The first in which is not limited to Africa, but when you compare Africa to the rest of the world, for example, we do find that there is inconsistent and a lack of funding for various different types of neuroscience, particularly for comparative neuroscience. There's basically nothing for comparative neuroscience. And often infrastructure is very poorly developed. Again, I'm speaking about as overall, not very specific places that where they do have some of the universities, particularly where I work in South Africa, University of Cape Town. There's a neuroscience institute that is quite advanced and does a lot of very cool things in terms of imaging and looking at real-time data in patients from MRIs and the likes. But that's a very costly experiments, and most 99% of African countries will not be able to afford that. So funding and infrastructure are two critical things that pose as significant challenges to continuing and providing more depth and more diversity of neuroscience across the African continent. Another thing that is a bit of a challenge is what is referred to as brain drain. And I use that word quite ironically because we're trying to study the brain, but what actually ends up happening is that people, and it's no fault of their own, but people qualify in Africa and then get jobs outside, maybe in the US, maybe in Australia, and never really come back. So we lose a lot of professionals in that way. And that's largely got to do with politics of the continent, and we're not here to talk about that. But it is a problem if people are being trained within the African continent and are being lost elsewhere on the globe. And so it does create this sense of a void and not many people to continue some of the work that has already been started or to continue the. So the last thing that has been identified as being problematic and challenging in Africa. Of course, there are many more, but I'll talk about these three in particular. Then the last of which is cultural clashes. Now, Africa is a very diverse, well, is incredibly diverse culturally. It varies significantly from north to south, from east to west, from the center to within a country. There are various different types of cultures and creating a sense and trying to do neuroscience amongst that cultural diversity can pose a bit of challenge because not everybody will understand things in the same way, and it's trying to overcome those hurdles that make it a bit difficult to continue work in African neuroscience, for example. Right. Now, I'm going to try and challenge you a little bit in this. I've talked a lot about, thinking differently, trying to have a different approach to neuroscience. And I'm going to pose well, I won't say a challenge, but I'm going to pose something to you that might take you a little bit back, but I want you to bear with me for a second, okay? So I'm going to talk a little bit about bats. And I know from COVID time, and particularly as Corpus Curiosum, was initiated during COVID, we know that bats through COVID got a really bad reputation. There were lots of ideas thrown about where the COVID pandemic came from. One of which was that it was a zoonotic virus that came from bats, and ever since bats have been deemed as quite evil and the likes. So anyway, but if we put that aside, I've worked with bats before as part of my career. I find them fascinating for many different reasons. Firstly, the idea that bats are the only mammals. Well, they are actually the only mammals that have true flight. There are no other mammals that fly around. If we look at birds, which are a different vertebrate class on its own, insects are completely different. But birds and bats are the only mammals that fly, flap wings and fly, which means that their bodily functions have to be adapted to this lifestyle. And I'll talk about that in a little bit more. But first, I want to talk about the controversies I put in here. We know about COVID. I won't mention that. I thought it was interesting. But what I really want to focus on is the phylogeny of the bats. If we look at the bat order, for example, for many years it has been a monophyletic order, in the sense that both megabats and microbats have been lumped together as just bats. Or the term for them was chiropterans. And you get megachiropterans and microchiropterans. In other words, the megabats and the microbats, the large bats and the small bats, is essentially what we are referring to in this. But when you look and you compare the megabats and the microbats to one another, and indeed to all other vertebrates, you find something very interesting in the body, and particularly in the brain. And what you do find is that the brains of megabats are very similar to that of primates, almost an exact carbon copy. Similarly, the brains of microbats are very similar in structure to insectivores. Now, I want to focus on this relationship here, because if the megabat brains are a good neuroanatomical duplicate of primate brains, well, this is an interesting proposition, or an interesting situation, because should we not think about using the megabats then as a potential candidate to understand how human brains function? Okay, they have a much closer evolutionary origin to humans and primates, as opposed to the rats and mice. So the time between the last common ancestor is much shorter from the megabats and the primates and humans versus humans and rats and mice alone. Okay, so if we look further into how bats operate and how they live their lives, what we find is a stark and a very interesting feature, is that bats tend to show a reduction in the amount of inflammation that they have. Why? What does this mean? So if we look at the life of the bat, it flies around, like I said before. There are many different things about its lifestyle that facilitate an improved and an increased immunity and an increased longevity. And in doing so, what you find, the overall effect is that there's a reduction in the amount of inflammation that you find in bats. And that's true of the body and the brain itself. We know that most diseases, whether it's neurodegenerative diseases or any other disease in the body, is largely the root cause is inflammation. So if we're trying to understand a model, would this not be a good model, the bats, as an alternative model, a closer model to understanding the effects of the onset, the epidemiology, the mechanism of diseases in humans, or use them as a model to translate to humans. So I think that we need to rethink how we are approaching our neuroscience, and I'll end off by just mentioning that again, there's an absolute wealth of diversity in Africa in terms of the faunal species that we do find here. I haven't even talked about the floral species, or indeed the fungi and the bacteria yet. So there's a rich diversity. Biodiversity is what I'm saying. And perhaps we need to rethink how can we utilize that effectively to perhaps further our understanding of what disease is in human brains, or in just disease in brains, for example, or just what is the variation of neuroanatomy across vertebrates, and maybe we'll uncover something that is similar or not that we can use to understand disease in a much better way. I think that there are lots of opportunities. I know that some people already have collaborations with people in Africa, and but perhaps this angle could foster new collaborations that everybody can benefit from. And so I think that's a really important angle that has never been truly has never really reached its potential as yet. And the last thing, and I think you probably, probably maybe a little tired of me saying this, but I will just say it one last time, is that we perhaps need to rethink how do we approach models that are used for neuroscience research. Is it possible that a comparative approach might yield something different or something beneficial? We won't know until we actually try. And we've done a lot of work with this, but we can't do it all by ourselves. And so there's, like I said, so many different species. It's lifetimes of work for many different people to continue all of this. So in other words, there's a lot of opportunity, and I hope that one day we would seriously consider it. So that's it for my talk. I hope I didn't take too much of your time. And I'd really like to open the floor for questions and discussions now.